Because now it is 71.
It's okay.
Yeah, I'm ready. I'm ready. We can.
Yeah, we'll start. Good morning, ladies and gentlemen. This is Relation. I, the member of Managing Committee of the Association of Fungal Biologists. Thank you for joining in for today's talk by Dr. Sanjeeva Nayaka. This is the first talk under AFB ILS talk series. ILS stands for Indian Lichenological Society. AFB stands for Association of Fungal Biologists. So this is the first talk under the collaboration between these two organizations. I would like to thank Dr. Sanjeeva Nayaka for agreeing to give the first talk under this talk series. Thank you very much, Dr. Sanjeeva Nayaka. Welcome to you to this program. I'd like to invite Dr. Sunil Kumar Deshmukh, the President of Association of Fungal Biologists, to say a few words on this occasion. Dr. Deshmukh, please.
Yeah. Good morning and welcome to AFB ILS this program. And today I will be talking something about my. Hello. Can you hear me?
Yes, sir, we can hear you.
Good morning and welcome to AFB ILS pre-webinar on lichen in extreme environment by Dr. Sanjeeva Nayaka from NBRI Lucknow. I will tell something about the Association of Fungal Biologists. The Association of Fungal Biologists is a scientific body with a broad interest in advancing mycology and cater to the need of mycologists worldwide. The association was formed to bring together mycologists with broad objectives of promoting and disseminating knowledge on different phases of mycology from wide perspective. The major emphasis has been laid to application of mycology to harness the biotechnological potential of macrofungi as well as microfungi for human welfare. We have over 250 life members, including professionals, budding mycologists, students with various interests in and capacity in fungal taxonomy, fungal physiology, ecology, pathology, phylogeny, genetics, molecular biology, forestry, biotechnology, and other allied fields. It also fosters collaboration, the exchange of ideas, and exploration of innovative approaches by studying and harvesting fungal's potential. This helps advance our understanding for fungi and their significance in addressing global challenges, fostering sustainable practice, and promoting overall well-being. This is a platform for networking, opportunities, helping disseminating cutting-edge research, and inspiring the participants to contribute to mycology. We had first AGM of AFB in December 23 at Mumbai. Second was recently held at Pune in October 24, and next is scheduled to be held on December 2025 at Kolhapur. The society is not only restricted to the Indian subcontinent, it extends its invitation to international mycologists to join the society for furtherance. You are invited to join the Association of Fungal Biologists and take advantage of webinars, conferences, and other activities. Today's webinar is important simply because it will give an idea about the lichens from extreme environment which can be used or harnessed for the biological applications. Over to Dr. Shinwat.
Thank you, Dr. Deshmukh, sir, for your kind words, for your guidance, and for motivating us to continue to do this kind of online events for the benefit of mycologists from the world. Now I will take a couple of minutes introducing today's speaker, Dr. Sanjeeva Nayaka, to today's audience. Dr. Sanjeeva Nayaka is a well-known figure in the field of lichenology. He's a lichen taxonomist. He's from India. He has more than 25 years of experience. He started his career as a project assistant, then soon joined as a scientist at CSIR National Botanical Research Institute, Lucknow. Currently, he is the chief scientist and the head of the PDSH division at the same institute. He had extensively explored different phytogeographic regions of India. He has published several floristic accounts, revised a dozen of taxa, discovered 53 new species, and reported over 176 species which are new to India. He had also carried out biomonitoring, biodeterioration, bioprospecting studies on lichens. Dr. Sanjeeva Nayaka's credibility can be assessed from his publication in recent in reputed journals. He has published around 240 papers. He has guided 11 Ph.D. students and has handled 14 externally funded projects as project investigator. Dr. Sanjeeva Nayaka has contributed immensely to the growth of lichenology, which is otherwise a neglected group in India. He established the Indian Lichenological Society (ILS) as a founding secretary. He has spread lichenology all over the country by training many research scholars, faculties, over 75 in lichen taxonomy. He has organized national and international conferences, so far 6 of them, workshops and training programs, around 20 of them so far. In addition, he has delivered over 60 talks and published popular articles in regional languages. I once again welcome Dr. Sanjeeva Nayaka to today's program. I request him to talk about Indian Lichenological Society before he starts his presentation on lichens in extreme environment. Over to you, Dr. Sanjeeva Nayaka.
Thank you, Dr. Shenoy. And also I thank the office bearers of AFB and ILS for giving me this opportunity. Indian Lichenological Society was established in the year 2014, just 10 years back, but soon it has got the status of a national association, and it is one of the very active association of the society which is promoting lichenological research in India. And over the period of time, we have experienced that the society also needs to entertain or promote the other cryptograms, because several cryptogram researcher has started approaching us. That's why today Indian Lichenological Society, along with the lichenology, it also promotes other cryptogramic research. And we have around 250 life members, and we every alternate years we keep organizing the conferences. And so far we have organized 5 major conferences, and we also have a journal that is called Cryptogram Biodiversity and Assessment, which is a biannual journal. And we overall look to promote the cryptograms and even the other parts of science also. And if anybody is interested to join the society, you are most welcome. So this is about the Indian Lichenological Society. And today the topic of my talk, as you all know, lichens in extreme environment. So you people might have seen the colorful lichens in the hilly areas. You people may be traveling sometimes to Himalayas or the higher altitude places in the Western Ghats or so. You might have seen these colorful lichens. Some lichens are bright orange in color, sometimes they're reddish, and sometimes yellow. You might have wondered why these lichens are such a bright colored ones. So you will get the answer to your queries in this talk today. So be with me till the end. So to begin with, it's important for me to give a kind of definition for the lichens. I'll not go deep into the definitions, but to give a brief introduction regarding the lichens. You people might have seen these kind of lichen thallus, lichens, grayish in color. So you know that lichens are a symbiotic association between fungus and algae. And if somebody asks you where is the algae here, where is the fungus, and this is how it looks. The whatever you look from outside, it is fungus, and lichen, that's algae is actually there in the inside. So see, if you even cut a section, you will see that whatever you see from outside, the upper side you see, this is the fungal part. Algae is there in the inside. So algae you don't see it from outside. So this is an intact association like that. So lichens are there on the earth since time immemorial. You don't know when it is there, but as per um, the fossil records, that is the two well-preserved fossil records, at least 415 million years ago, they have came into existence. So, but they are introduced to the pharma world by the name lichens by Theophrastus in 300 BC. And you see the time gap here till 1867. People, they didn't know that lichens are actually made up of two organisms. They thought that it's a single organism. It is a separate group for classification, separate taxa altogether that considered. But only in 1867, Simon Swidner, a Swiss scientist, proposed the theory that lichens are made up of actually two organisms, that is fungus and algae. And these are some of the drawings, his preliminary drawings that he has made. But of course, he faced a lot of criticism in the beginning. And only after a century or so, that concept of dual nature of lichens, or the two organism in lichens, was accepted. See again, it took almost a century for accepting the concept of dual organism nature of lichens. So then meanwhile, over the period of time, several other organisms well associated with the lichens are discovered. It may be the cyanobacteria or the other algae, or the other fungus living with association with lichens, bacteria, yeast, protist, and virus. Even some of the microinvertebrates are also found in association with the lichens. So now this association is not a simple association. Most of the time, this association is very unique and very specific. So each organism associated with the lichen have some role to play, whether it may be in the nitrogen fixation or other nutrients absorption. So these organism has a very specific role to play in the lichen association. So that is why the definition of lichens kept on changing. In the beginning, when Swidner proposed a dual nature, it is called as lichens as symbiotic association between fungi and algae. Then the definition kept on changing, and since today we know that lichen is not association of only two organisms, there are many other organisms also involved. The definition is like this. A lichen is a self-sustaining ecosystem formed by interaction of an exhabitant fungus and an extracellular arrangement of one or more photosynthetic partner and an indeterminate number of other microscopic organisms. So in brief, we can say that lichen is an microecosystem where fungus and a photosynthetic organism act as a primary symbionts, whereas the all other are the parasymbionts or the secondary symbionts. So then because of this symbiotic association, lichens gets several unique characteristics. So otherwise, fungus or the algae alone might not have got this much of potential or the ability to perform in the nature. So because of the association, lichens is a very strong, very strong organism which can have special characteristics, like they can grow on all kinds of substrate. Maybe it may be rock, because I say rock here because it is one of the hostile environment. It doesn't have any nutrition, so lichens can grow comfortably on the rocks, moraine, concrete, timbers, all the building materials maybe, and also on the tree bark and all. So it can grow in the harsh and the hostile environment. So the environment where most of the organism cannot survive, these lichens will easily survive. So that are the extreme environment. But they're slow growing, their metabolic rate is poor, they're slow, and also they're poor competitor. Unlike other fungus, these lichen fungus cannot compete with the other fast growing ones. So they are poikilohydrous, means they cannot control their water balance. So when the water is available in plenty, they take the water, and when there is a drought kind of conditions is there, they lose all the water and remain inactive. So that is why they are desiccation or they drought tolerant also. They remain physiologically inactive during the harsh climatic conditions. Lichens are very extremely tolerant to the freezing stress and low temperature. People have seen that they could withstand temperature as low as minus 196 degree. They have survived for 96 weeks in a frozen condition, that is at minus 15 degree for 10 years. 96 weeks in 15 degree, minus 15 degree, and 10 years in minus 20 degree. They have high content of the carbohydrates of the polyols, which are speculated to be reason for their high tolerance to the freezing stress. Frost injuries are never seen in the lichens when they're growing the natural condition. They can photosynthesize in the low temperature or the reduced light. So they found a photosynthesizing even at minus 20 degree, even under the snow. Another important thing about lichens is that because of this symbiotic association, they produce over 1,000 unique secondary metabolites. These secondary metabolites are found only in lichens, not in any other group of organism. That is one of the unique characteristic of lichens. Now coming to the extreme environment, what is an extreme environment? The harsh condition, habitats with a harsh condition that make it difficult or the impossible for most of the organisms to survive. So this is the harsh conditions, harsh extreme environment. Of course, you might have seen the several extreme environment condition. Maybe it may be a geothermal water or the hot spring, what you say. There also you will get sometimes algae and the microbes. So for most of the organisms, that is in harsh conditions, but some organisms live. So the habitat with the harsh condition that makes it difficult or impossible for the most of the organisms to survive is called as extreme environment. It can be extreme temperature, it can be cold, extreme cold, or the extreme heat, or it may be high pressure, as you see in the deep sea. It may be salinity, it may be hypersaline environment, or pH may be extreme. It may be acidic, less than 5, or alkaline, more than 9, or it can be high radiation, high bright sunlight, along with the UV radiation. It can be dryness, very arid environment. So you will see some of these extreme conditions in regions like polar region, like Antarctica or Arctic. You will see it in the hot deserts, deep sea, and as I told you, hot geothermal springs, or you can see it in some of the top of the mountains, maybe in the Himalayas, like Everest, which is the highest mountain. You can see such conditions in the volcano or the upper atmosphere or the outer space. So these are some of the examples for the extreme environment. But for today's talk, I have selected only cold deserts because I have first-hand experience in working in these cold deserts. So that is why I will be restricting to the cold deserts here. Whereas in the other conditions, maybe in the geothermal springs, the lichens are not found, or of course lichens are there in the high mountains, but they have faced the similar situation as in the condition of the cold environment. So so coming to the desert, the cold desert is a region that has less than 254 millimeter, that is less than 10 inches of annual rainfall or the precipitation is called the desert. In the cold desert is apart from the less precipitation, it also has the cold conditions, the extreme cold. That is why it is called cold conditions. So usually in the desert or with the cold desert or the hot desert, the humidity is very less. So in Antarctica, for example, you see rainfall precipitation is just 50 to 20 or 50 to 200 millimeter, whereas the temperature can go as low as minus 89.2 degree. So this is the map of Antarctica. I had been to Antarctica twice. So this is Antarctica, and this portion you see, the tail kind of portion, what you see here, this is called Antarctica Peninsula. This is much warmer here, this area, whereas the other region is very much cold. So this is called maritime Antarctica, and this is the Antarctica Peninsula. So then if you're coming to the other cold desert in the world, there are several places. cold deserts are there. But India has the two popular cold deserts in the Western Himalaya, that is Leh and Ladakh, and another one is Lahul and Spiti. So the Leh Ladakh is in Jammu Kashmir, whereas the Lahul Spiti is there in the Himachal Pradesh. So here the rainfall or the precipitation is less than 100 millimeter, and temperature can range from -40 degree to +40 degree. Sometimes in the Dras, there is a place called Dras where the temperature has been measured as low as -59 degrees centigrade. So then this is the cold desert the region map you can see for India. Like Kargil, I have got an opportunity to go up to Kargil, Leh Ladakh here, and below here it comes the in the Himachal Pradesh is comes Leh Ladakh. So these some of the common features of the cold desert, whether it is in India or Antarctica. One thing is you can see desert. This map this picture you can see there is no vegetation, or some vegetations you can see here. They're very bushy kind of plants. I'll show in the next slide. So this is the clear picture of a cold desert. You can see at the background the snow mountains are there. And this is again a Ladakh picture where you can see barren land with the snow caps. This is Antarctica again. You see in the Antarctica, you will see the barren land here, no vegetation at all, and only soil or the rocks you will see, and in the background you will see ice here. So here you can see this is a picture taken during the winter. Here we have our station Maitri here, and in the front you will see a lake. There is again frozen now in this picture, and only ice you will see. And there is in Antarctica only 2% of the land is free of ice, and where in that place we have the station, and that place we go for our scientific study and plant collection and all. So then what is the extreme environment at the cold desert? One thing is that low humidity. So in Ladakh has the humidity which is up to 40 to 50%. In Antarctica, the humidity is very, very low, just 5 to 9%. Average is up to 0.03%. So you can imagine how harsh the climatic conditions there, humidity is there. But these mountains have the high radiations. They have bright sunlight combined with the UV radiation. And apart from that, in this area, wind is very, very strong, and they're erosive wind. In Ladakh, wind speed can go up to 40 to 60 km/h, whereas in Antarctica, the wind speed may go up to more than 300 km/h. So you can, if somebody had gone to Leh Ladakh area, you might have seen this kind of pictures, this kind of landscapes. They're called earth pillars. So sharp pillar kind of things you will see. These are the sand pillars, and this kind of structure has become mainly because of the wind. So erosive wind. So because of the wind, the soil has eroded, and this kind of pillars have formed. So this kind of structure also sometimes you will see in the Antarctica. So in the cold desert, you will get a very short period for the growing season. Very active metabolite season is very less, because if you take Antarctica, another harsh condition is that there the 6 months day, 6 months night condition you will see. So at least in a year you will get 2 months of continuous darkness you will get. So and it is followed by the snow cover and all. So only in the summer, few days or the few months, the ice cover will be melted. So then during that period, these plants there get a season for their active metabolism or the growth. So even in the cold deserts of in the Western Himalaya, Laul Spiti or Leh Ladakh, see most of the time it is cold, or the long duration of snow cover limit the period for the growth. And this, as I told you, there is a long-lasting snow cover will be there. Soil is frozen, or permafrost we call, and there is no other vegetation. The minimal vegetation is there, like lichens, which needs which needs other plants for their support to grow. There will be no availability of the substratum for the lichens or the other epiphytes, and in these areas there will be dominance of the cryptogams, especially lichens. You will get it. So sometimes you'll get moss and other bryophyte, but lichens are the prominent cryptogam that you'll see in the cold deserts. And so again, the picture of the Ladakh here, you see, these are the, as I told you, the earth pillars here. And in Antarctica, this is what I'm showing here is the effect of the wind here. This, as I told you, these earth pillars here. And in Antarctica, whenever there is a strong wind, it leads to blizzard. We say blizzard it is. So it comes along with the snow, lots of snow. Here the picture, it is of snow blizzard here, and this is the picture taken after the snow blizzard. So you can see the how much snow has been accumulated, and sometimes the snow blizzard in Antarctica can go for hours together or days together, leading to the whiteout. So where you will cannot see anything except whiteness outside, which leads to the loss of your route or the way if you're not experiencing such an environment. So vegetation, if you take in this harsh environment, this is a picture of the cold deserts in India. You will get a bushy plants like this. This is a karagana, and it has a deep root system. The roots goes deep into the soil, but from outside it is bushy like this, and also it is thorny. Prickles will be there in most of the plants there. And other vegetation, as you see, lichens here, you will get bright lichens here, bright colored lichens here, mostly growing on the rock surfaces, very rarely on the soil. And of course, since there are no higher plants, or the trees kind of plants are very less there, and you will not get much lichens as an epiphyte. And this is the cold desert excursion that I made during the year 2011 to the Leh Ladakh here. This is the route we followed. We started from Manali and then Rohtang Pass and all. So we went up to, as I told you, Kargil, Dras here, Zojila Pass, and finally came to Srinagar. So we started from altitude of 2050 meters, and then we reached the maximum altitude of 5,602 meters, and then came back to the 1,585 meters in Srinagar. So these are some of the pictures of the cold desert excursion. This is at Manali, first picture, that is near the Hidimba temple in Manali, and they can see some of the altitude mentioned here, or the for example, altitude in Patiso, it is 12,300 feet here, and the cold desert already has started, you can see here. And some of the prominent landmarks in the cold desert excursion you will get, Buddhist monastery and all, and you can see here Kargil War Memorial, Tiger Hill, and other things here. And in Antarctica, so Antarctica, I had been to Antarctica twice during the 22 and 28th expeditions, Indian Antarctic expedition. And Antarctica, geographically, that's the South Pole. All the area below 600, 66 degree 33 minutes of the south latitude is called as Antarctica. And if you take an area, it is 14 million square kilometer. It is larger than India and China put together, but it is 97.6% is permanently covered with ice. Only 2.4% is free of ice, and that is the place where usually people go construct their research station and other things. And other important thing in Antarctica is that world's 90% of water is frozen, and if it can be melted, it can be supplied to the 75% of the world. And it's just a speculation, Now, what I mean to say that 75% of the world supply of fresh water is there in the stored from in the ice from in Antarctica. So if at all in the future due to the climate change or sudden rise in the temperature takes place, so all the ice in the Antarctica melts, it can rise the sea level to 60 to 65 meters. So some of the facts of Antarctica: it's a coldest desert, coldest place on the earth, minus 89 degrees the temperature, and the average temperature you can have is that 1 degree centigrade. It's windiest, it's the highest. So if you take the average height of the Antarctica, it is 4,000 meter, 4,897 meter, and the highest place, of course, it's very less, 2,160 meters, but an average height is 4,897 meters. It is driest, humidity is very less, emptiest, it is no man's land. People habitation is not there. Human habitation is not there. Only people who go for research purpose or the adventure purpose, a tourist, they stay in Antarctica for few months and they come back to the mainland. As I told you, it is a place where the 6 month day and the 6 month night is there. And that is because of the harsh climatic conditions there. The Antarctica gets several names, like it's a continent of extremes, the icy continent, the frozen continent, land of penguins, residence of blizzard, seventh continent, white continent. And these are some of the names that is given to Antarctica. And Antarctica is a place where the ozone hole has been detected. And in the 13th September 2007, NASA took this image where the largest ozone hole, that is 9.7 million square kilometer area of the ozone hole could be seen at that time. It is discovered in the ozone hole was detected in the 1970 by British Antarctic Survey scientists. And the hole means there's not an exact hole there. The only thing is that ozone layer has been thinned down severely. So you may be knowing the different layers of atmosphere here. So the ozone layer is there above the stratosphere, and which has depleted due to the CFC, that is carbon fluorocarbons, which is emitted by the pollutants that has gone up to that level and depleted the ozone layer. So the Antarctica is a place where the several climate change studies are being conducted, and it's an ice in the Antarctica are the indicators of the global warming. For example, this Dakshina Gangotri glacier, it is there near our station Maitri in the Shirmakar oasis in Antarctica. So this the V kind of thing, the white point, what you see, this is called a snout of Dakshina Gangotri. What it does in the winter, because of the snowfall and all, it advances, it comes forward, it grows, but during the summer, it melts and goes back, retreats. So the people usually measure this advance and the retreating things, and it is now observed that because of the climate change, the snout has retreated much backwards. So apart from, you can see that the collapse of the Larsen B shelf in the 2002, this has been nicely captured by the satellite. So you can clearly see, because of the climate change, the ice is melting, or the ice shelf is getting broken in Antarctica. So other important thing on the Antarctica, you will see beautiful sky here. This is called auroras, aurora australis. The flares of colors on the sky. It's a very special phenomena. And other thing is that the water around the Antarctica continent, the water in the Antarctica, it gets frozen during the winter, and summer it melts. And that is the time during the summer when the ice melts, the people visit Antarctica by ship. So otherwise, these very thick ice sheet do not allow anybody to visit there. Certainly not possible by ship. So because it in the winter it gets frozen, and in the summer it gets melted. So the area of the Antarctica gets stretched and is shrunken. That is why it is called as pulsating continent. So you can see sometimes the ice can get frozen up to 1,000 kilometers from the border of the continent. So, so these are the Antarctica continent. There are several stations are there. 30 countries have constructed their stations around the Antarctica, and among them 46 are very active. Um, um, and 30 fields are the small camps are there, and summer population is around 4,200. In the winter the population is 1,150. In the winter the people who will be doing some kind of experiment during the winter, or who people who are maintaining their station will remain there. Otherwise during the summer, after the completion of their work, they come back. So when here it is winter here in the India or in the North Pole, it will be summer in the South Pole, in the Antarctica. Now it is summer in Antarctica, and this is the time when the people go there. And this is the station, this is the research organization, National Center for Antarctic and Ocean Research. Of course, now the name has changed. National Center for Antarctic and Polar Research it has become, and this is the organization which conducts expedition to Antarctica. So there are several procedures for that. If somebody is interested, I can tell you later. So as I told you, I've participated in 2 Antarctica expedition. This is the route we follow from Goa or Mumbai. Earlier people used to go to directly by ship to Maitri station here, one stop at Mauritius, and that is covering 11,000 kilometers. They used to take 35 days. Later on, the expedition route has been changed. This reaching Antarctica route has been changed. From Goa or the Mumbai, people will go to flight to Johannesburg to Cape Town, and from Cape Town, the people catch the board the ship, and from Cape Town to Maitri Antarctica, it would take 10 days and 4,112 kilometers it used to take. And nowadays from Cape Town we have flight to the area where our station is there, and it would take just 6 hours. So this is the improvement in over the period of time in the Antarctic expedition conducted by India. So this is when I went to Antarctica by ship. One ship was the Magdalena Oldendorf, another was the Emerald Sea, and this is while going by flight. You can see that this is IL-70 flight. It's a cargo flight actually, and it is modified for the passenger. And we have in Antarctica behind our station the airport, the runway on the frozen ice. So it's a beautiful experience. And this is our ship and the other vehicles in the Antarctica. This is a small flight for the carrying the people from this station to the nearby stations. And these are some of the pictures of the Antarctica ice. While going to Antarctica by ship, you will get different shapes of ice and all. So this is our first station, Dakshina Gangotri, which is built in 1983 on the ice. And now this is completely sunken in the ice, and you don't see them. It is completely buried. And this is the station Maitri in Antarctica, and this is a winter station. Actually, you can see it is built on a raised platform here, and it has accommodation for around 30 people and all facility we have. And these are the summer camp. Here the people usually go for the wintering, they stay here, and the people who go to summer time only, they stay in this kind of accommodations here, container modified into living modules here. And we have the another station called Bharti that is built in the Last Man Hills here. This is the Last Man here. This is the place where the Last Man Hills are there, and there it is a modernized station has been built. And if you see the biodiversity in Antarctica, there are 9 penguin species are there. Out of the 17 species, 9 species of the penguins will get in Antarctica, and out of 35 birds are there, and there are some seals and the other species of the mammals are there here, and these are the whales. And Antarctica other biodiversity, this is the largest insect there in the Antarctica, Belgica. There are very few species, but this is the largest one. And these are the only two angiosperm plants you get in the Antarctica, that too in the Antarctica Peninsula. These are the only two native plants. Otherwise, you have several other species of plants which are just exotic for the Antarctica continent. And if you see Ladakh, again a cold desert here, we have around 750 plant species here, 540 dicots and 65 monocots and 2 gymnosperms. And see, the main floristic components are the cryptogams, as I told you, algae, around 700 aquatic and terrestrial, 700 seaweeds, fungi around 100 species, bryophyte around 140 species, and lichens are the dominant in the dominant number, around 500 species here. And the lichens, that the diverse group there, and a total of 40% of the lichen species are endemic to Antarctica, 25 species are restricted to southern hemisphere only, 100 species are bipolar, they are found both in the North Pole and the South Pole, and 80 species are cosmopolitan. So these are the lichen exploration site in the 3 expedition. Before me, Dr. Upreti and Dr. Vivek Pandey had already visited Antarctica and collected the lichens. And finally, I got an opportunity to explore the whole of the Schirmacher oasis for lichens. And if you compare to the lichen diversity in the cold desert in the India, so Indian cold desert, we have around 121 lichen species. As Antarctica as a whole, 500 species, Ladakh 75, Lahore Spiti 79, and Schirmacher oasis have 69 species, where our Indian station is there, where I had collected lichens. Where the Bharti station is there, that is called MacLeod Island in the Lastman Hills. So there are 25 species of lichens are there. And these are some of the lichens of the Indian cold desert. You can see the colorful lichens here. This again the lichen pictures in the cold deserts. So these are some of the other interesting thing is that in the cold desert, the bright colored lichens ones belongs to the family Telostaceae. They are more common in the cold desert. And see the how the lichens are survive in the such a cold conditions. How do they adapt themselves to the extreme conditions? So one thing is that the lichens requires a very little rainfall to survive. They don't really depend on the rainfall. The less humidity or the snow meltwater is sufficient for their survival. Their thallus is hygroscopic, as I told you. They are picolohydric plants. Their thallus is very much hygroscopic. They quickly absorb the moisture from the atmosphere or the snow meltwater, and they start their metabolic activities. And the rhizines are there. The rhizines with the root kind of structures are there in the lichens, but they're only for anchoring. Otherwise, their whole thallus is very much responsible for the absorption of the water. So see this picture. This is very peculiar, and this is what you have to appreciate in the lichens growing in a very extreme environment. So the one thing is that they grow on all kind of substratum. They grow on solid rock. Moraine is that permafrost, or the moraine is the soil that is mixed with the rock particles, and they grow on concrete and other things. Here you can see they prefer to grow in a site that provides heat or facilitates snow melt water. You can see this right extreme picture here. At the upper portion, there is a snow accumulation is there, and it melts and the water channel has formed. You can clearly see here, and the lichens are growing in that area. So lichens usually prefer growing in such crevices, such water channels. Here you can clearly see here. This is how it made. Here you can see the black things here. These are actually lichens. So it is actually a passage or the gap area between the two lakes. In the upper there is a lake here. In the below there is a lake here. I don't have the complete picture. It is not possible to click. And when the upper lake, it gets filled with the when it get melted, and also it gets the water from the shelf ice here. I mean the ice here. It gets filled, and water overflows through this channel, and it fills the next lake which is there below. And in this channel, these lichens are growing. And it also uses the snowmelt water, which is covered on over them. And it also depends on the water that is coming from the upper lake here. So this is a beauty, how nicely they have adapted to this condition for the sake of water here. And the most of the lichens in Antarctica are crustose. Crustose lichens are very sturdy lichens, very small in size, smaller in size, very stunted, clumped. They're closely aggregated, and that is how the crustose lichens are more dominant. And because of the heavy wind, the folios of the fruticose lichens can be blown off, whereas the crustose lichens stick very much attached to the rock surfaces here. Some of the lichens, as I told you, are still having the clumpy nature with the stipe and the bulged growth they will be having. So that will be helping observing the more moisture and retaining it. So some of the lichens in Antarctica are endolithic lichens, means they appear from outside like only in the fruiting body. This is apothecia. You'll see only apothecia. You're not seeing the thallus here. The thallus is actually there inside the rock here. So this rock may be porous or the loose ones. The lichens you from outside you will see as a only small dots like this, and the thallus are there in the inside. This is another adaptation. So, and again, sometimes the lichens are found in the gaps of the rock or the boulders or the crevices or depressions. So the cracks or the crevices in the rock, it helps in capturing the lichen spores, one thing. Second thing, it also holds some soil particles as well as moisture. So in that condition, lichens can grow easily, as well as it will help them help the lichen to anchor very well to the substratum. It don't get easily blown off by the fast wind. So this is the other adaptation of the lichens. So another thing is that most of the time lichen prefer to grow opposite to cooling wind, opposite to the cooling winds. If there is a rock or a slope kind of structure, things are there, it grows on the opposite sides to the wind. So it will be protected by the severe cold wind. So that is another adaptation. So the lichens, again, you see the lichens, the rock usually do not have any nutrition, but although lichens have the very less demand for the nutrition, but sometimes they prefer to grow where they get the nitrogen source. For example, penguin rookeries. So here the penguin rookeries are there, and penguin rookery or the penguins are one of the important organism in Antarctica for fertilizing the soil, whether it is for lichens or the other plants. So it's an important source, nitrogen source. Nitrogen is important for plant growth. You can see in this picture, the rookery of the lichens are the penguin is there here. Apart from this area, the nitrogen get volatilized, and it also goes to the upper areas here, and that is how finally the nitrogen gets into the plant's body or the soil and increase the fertility. So lichens prefer to grow in such areas, the nitrogen rich areas. And as I told you in the beginning, some of the lichens in the Antarctica are the dark colored. It may be yellow, red, brown, or black pigmented. So dark colored lichens, there is a very important role to play. Color, you know that black bodies absorb more heat. So that is what happens here. The lichens like black in color, so they absorb the sunlight very immediately, and it gets warmed. And because of when it becomes warm, so it melts the ice, the snow on its cover, and it gets water, as well as thallus also gets warm, and it immediately starts its metabolic activities. So that is why in Antarctica you will get these dark colored lichens, whether it's Antarctica or in the cold deserts, especially black ones. So apart from that, the bright colored lichens, what you see, they have the pigments, whether it is anthraquinone or atronorine or acetic acid. These are some of the secondary metabolites, they're produced by the lichens, they're deposited on the upper cortex. So it becomes translucent. So the sunlight has to pass through the upper cortex to the algal layer below. If you remember the first picture, what I've shown, the section of the lichens where the lichens algae is there inside the fungus thallus or lichen thallus. So the light reaching to the algal layer is reduced in quantity as well as in the harshness, because the pigments, the color filters the light which is going to the reaching to the algal layer, because algae is one which is sensitive. Algae is the one which synthesizes photosynthesizes and prepares carbohydrates. So that is why pigments have a very important role to play. That is why lichens in the cold deserts are very bright in color. So the lichens also produce mycosporin-like amino acids, which are capable of absorbing the UVB radiation. It is well worked out, and the mycosporin-like pigment, this one amino acids, they are very much helpful. They are produced by the lichens in the cold deserts. See, this is the section of the lichen thallus. You can see again here, this is the same picture. Upper cortex is there, algal layer, and the other fungal components are there. And in Antarctica, it is seen that the lichens usually have a thicker cortex and a necrotic layer. It is called epicortex, or the necrotic layers you can find on the cortex of the lichens here. This is the epinecral layer. Necrotic layer is nothing but the dead algal cells or the dead fungal cells will be deposited above the lichen cortex, which again reduces the light passing through the cortex to the algal layer. And a thicker cortex and the epinecral also act as a barrier for the water absorption as well as the water loss. So it avoids like oversaturation of the lichen thallus and also the quick loss of the water. So so in the lichens in Antarctica, it is seen that their lichens thallus is thicker, but they are very stunted in growth, or they very less in the width of the thallus. So they're smaller but thicker in the shape and the thickness. So and again, one more thing is seen in the Antarctic lichen is that the mycobiont component is biomass is more compared to the photobiont. So in the lichen, usually only the 10% is made up of algae or the photosynthetic organism, whereas the 90 to 90% is of the fungal component. So fungus is a dominating organism in the symbiosis. So whereas in the Antarctic lichen, it has been observed that photobiont component is very, very less. It is sometimes 5 or less than 5%. Only fungal component is more. That is the one of the reason that why lichen thallus are very smaller in size, because algae is a one which produces carbohydrates, which is essential for the growth of the lichens. Then other things like tolerance to the temperature, extremely tolerant to the freezing temperature, as I already told you. And so why the lichens are extremely tolerant? Because they have high content of carbohydrates, polyols. So some of the polyols like ribitol, arabitol, mannitol, and trehalose, these are some of the polyols which are found in the lichen thallus, which help them in tolerating the freezing stress. So the also the several metabolites, as already told in the lichens, there are several metabolites. Sometimes a lichen can have up to 50-60 metabolites a single lichen. So these metabolites also act as an antioxidant agents. So they protect the lichen from the oxidation stress. So this is another thing. Then it has been observed that when the freezing condition comes, the ice nucleation takes place. The water freezes at the intracellular spaces of the cells, so not inside the cells. So because of this intracellular space ice freezing takes place, sometimes cytolysis takes place. Cytolysis is nothing but the collapsing of the cell walls. But this condition, it has been observed that it can be reversible. So once the favorable condition comes, the again lichen comes back to its original shape and size. And frost injuries is never seen in the naturally growing lichens. Other physiological activities for the lichens growing the extreme condition is that they can photosynthesize in low temperature. I told you. So and in the extreme conditions, as I've already told, that lichens minimize their metabolic activities, and the metabolic activities very is lowest just to survive. So so other here, I told you that the size of the lichens is very small here. See, another because see, it is not because of the only nutrition, lack of the nutrition, they are very small. Because what happens in the Antarctica, when the favorable condition takes place, immediately they start respiring, and because of the respiration they lose a lot of water, a lot of carbon. So before they could photosynthesize, they lose a lot of carbon because of the respiration. Even during the hibernation period or when they're inactive, respiration keeps continuing. There is a loss of carbon, but when the favorable condition comes, although photosynthesis takes place, that is not really sufficient for the carbon buildup. So carbohydrate synthesis. So the loss and the accumulation of carbon, there is a kind of imbalance, or the kind of insufficiency is there. That is why the lichen thallus are very smaller in Antarctica. This is one of the experiments which I had carried out. Of course, there are other experiments, but I'll not be showing that. So the poikilohydric condition of the lichens are how a lichen will come back immediately to its original condition. So what I did, I took two lichens, Umbilicaria ducicata and aprina. So I dehydrated the lichens, and you can see the water content in the lichens has gone down to below 10% here. So and after a long time of desiccation or the drought, when I gave water again, the water content in the lichens came back to its original status, and also the lichen become very active. So that is how the lichens are called as poikilohydric, or the resurrection plants. They come back to the original conditions. So these are two of the lichens from the Antarctica only that has used for these experiments. And how do the lichens reproduce in the Antarctica? Reproduction is mostly by vegetative methods. Either they produce by Isidia, Soredia, or thallus fragments, or sometimes through the pycnidia and all. So sexual reproduction is very much less, even though the fruiting bodies like apothecia and all produced, they most of the time bear very sterile spores. Either the apothecia will not produce any spores, or if they produce, they are sterile or the immature spore. So because the sexual reproduction is energy consuming for the lichens, to conserve the energy, they mostly reproduce by vegetative method. So summer is the better time for the growth and the reproduction. But the lichens in the Antarctica are very slow growing, but they can be very long lived. Longevity is very long. Some of the lichens in Antarctica or in the cold deserts can reach their age up to thousands years. A lichen in Alaska, that is Rhizocarpon geographicum, this lichen is very much common in the cold deserts, whether it is in India or the other part of the world, as well as in Antarctica. It is also called as map lichen. In Alaska, it has been measured that some of the lichen thallus are as old as 11,500 years. So lichens are some of the oldest living organism on the earth. And because of the sturdy conditions of the lichen thallus or the lichens, so they are also used for the several experiments, especially in the space experiments. In one of the experiments that is called as lichen and fungi experiment, that is LIFE, in the International Space Station, this lichen, the Xanthera elegans or Risvaschia elegans, was sent there, and it was kept in the outer atmosphere of the outer space for 1.5 years, and it is exposed to ultraviolet radiation, cosmic radiation, insolation, extremely low temperature, vacuum condition and all, and it is brought back to the earth, and it could be seen that 71% of the lichens remained viable. So of course, this is short duration, but it is lichens, 71% lichens were resisted this harsh condition there. So like that, the lichens have been also sent to the, they're not sent to the Mars, but in the same research center, that is International Space Station, they stimulated the Mars condition there, and they kept the lichens for the experiment for 1.5 years. So the lichens utilized there are the endolithic lichens, because what happens, in the Mars, if you see the condition there, the strong windstorm is there. So strong, apart from the cold temperature and the bright light and other things, wind is one of the major factor there in the Mars. That is what they simulated in the space station, and they utilized the endolithic lichens. So because the thallus is very less exposed there, only a few black spots you can see there, but lichens are there inside. You can see this picture, lichens are there inside the crevices of the rock here like this. So that is where they used it. So again, when they brought back to Earth, they could see that 60% of the lichens remained intact and also viable. So they speculate that endolichenic fungi in Mars may tolerate low temperature. In future, they can send the endolithic lichens to the Mars also. But all this is a very short duration experiments. Although these lichens are found adaptable there, but they require at least some short period of favorable condition for their growth. So unless the short condition is there, lichens won't survive. So in conclusion, that lichens are Grimm's stress-tolerant organism. Grimm is an author who said that Grimm's stress-tolerant organisms they have the slow growth rates, significant longevity, low demand for the nutrients, and adaptation to the stressful condition. They are very much adaptable to the stressful condition. So lichens are Grimm's stress-tolerant organisms. But as Dr. Deshmukh sir already told us, these lichens growing in the extreme conditions should be utilized for the betterment of the human life or the human kind. Maybe we can identify the cold tolerant molecules, whether it can be a secondary metabolites or the carbohydrates, or it can be a protein, whatever that is responsible for cold tolerance. It can be identified, and identification and utilization of the radiation filtering molecules can be identified and utilized. Maybe it may be pigments like anthraquinones, arsenic acid, and other things. Of course, such kind of experiments are already taking place. For example, with the help of our lab at DRDO, Leh, such experiment has been carried out to utilize the lichen metabolites in the sunscreen lotions or the sunscreen creams that has been being experimented, or skin ointment. So identification of the genes responsible for the cold or the desiccation tolerant, that can be identified. Such a genes, if can be identified and introduced into the plants or the transgenic plants, we have in India several acres of land which has the dry conditions. There we need plants which will survive in such a conditions and produce the food for the human beings. So these lichens has the very much potential for their utilization in future in the bioprospection area. But this has to be done carefully, because Antarctica is a place for research only, not for exploitation, and so that has to be done logically, keeping in the view of all the rules and regulations. So this is about the lichens which are growing in the extreme conditions. Thank you very much, and I'll be happy if you have questions to be asked. Thank you.
Thank you, Dr. Sanjeeva Nayaka. That was a wonderful presentation. Thank you for sharing your experiences for collecting and studying lichens from extreme environment like Ladakh and Southern Ocean or Antarctica. I have a question, if you don't mind. Can I ask that, please?
Yeah, please go ahead.
I would like to know if the lichens from the extreme environment came onto the Earth compared to their counterparts from the non-extreme environment.
No, I didn't get your question. Please, can you repeat?
I was wondering if the lichens present in surviving in extreme conditions, environments, they came onto the Earth before their counterparts from the non-extreme environments.
See, the extreme we have studied the species composition in the extreme environments, as well as the normal areas. See, there are some lichens, at least some genus are common between the extreme conditions as well as in the other areas, normal areas. So some of the lichens like Xanthoria, Rhizocarpans are cosmopolitan, not cosmopolitan, they are available in colder area also. See, only few species are endemic, are they are very much adapted to they are available only in these extreme conditions. So, but their counterparts are of the same genus, are they available in the other areas. So because of these areas, these extreme conditions, they have adapted themselves there and they're surviving better in such conditions. So this is just a kind of adaptation for their survival there. And if some species are common, they can be brought, but not all the species can be brought into the normal conditions and expect them to grow here. They don't survive.
Thank you, Dr. Sanjeeva Nayaka. Dr. Nivedita Pradhan, if you have a question, please do ask. Dr. Nivedita Pradhan. I hope you can hear me. Okay, Mr. Sachin Rajput, please ask your question.
Yes, sir. I want to ask these lichens growing in these extreme environments, they produce some unique biomolecules to protect them from the precipitation, basically light and all. Do they also produce some metabolites, secondary metabolites, which protect them from scavenging or predation by other organisms?
Yes, the lichens already they produce such kind of things. For example, we have seen the lichens having several metabolites, and we wondered why it is producing such a range of metabolites. One thing is for herbivory. Like the lichens are not eaten easily by the insects and the other organisms, even for the other herbivores, because these metabolites which are present in the lichens are bitter in taste, one thing, and also sometimes poisonous. So that is why they are not eaten by the herbivores. So similarly, the lichens growing in the extreme environment also have these metabolites inbuilt in them. So that is there. Thank you.
Okay, Dr. Venkatesh, please.
Sir, good morning, sir. I have a doubt, sir. In
Yeah.
foliage lichens, we can't see in all the plants. We are seeing only in the some plants only. Is there any molecular level inhibitors or any genes are involved for inhibition of the interaction of these lichens, sir?
That kind of studies has not been carried out as per my knowledge. See, the growth forms, that is folios, fruticose and all, no, they all depend on the climatic condition of an area. So if the climatic conditions, it can also be the altitudinal gradient or the place. For example, in a place where in Lucknow, which is plain area, altitude is very less, the climate is much harsher, or the temperature is very high, dryness is there. Here we don't get the foliose lichens, we get only the crustose lichens. But whereas in Bangalore, if you go to Bangalore or sometimes in Pune, you will get the foliose lichens. Although diversity is not much, but you will get the foliose lichen. Bangalore is good place for the lichens to grow. So it all depends on the climate of an area rather than the any genes to do with that. So and if you go towards the hilly areas or the Himalayas or the Western Ghats and you will get plenty of crustose as well as the foliose fruticose lichens also. And even otherwise, if you see the proportion of the different growth forms, crustose lichens are more in numbers than the foliose, and the fruticose are the very less. The proportion itself is less, and apart from that, this is the climatic condition is also important. Yeah.
Thank you, sir.
Thank you. Ladies and gentlemen, do you have any more questions? Okay. Dr. Shah, please ask your question. Dr. Dharmendra Shah, please ask your question.
Good afternoon, sir.
Yeah.
So, do we find lichens in extreme environments near rocks along the shore?
Yes. See, there are lichens which are reported from the coastal areas. Not many lichens, but there at least there are few species are there. For example, the different species of verrucariae are found along the coastal areas, either submerged in the water or sometimes semi-emergent. So even on the coastal area, even near the seashore, you will get lichens. There is absolutely no doubt you will get them.
Thank you, sir.
Ladies and gentlemen, if you have more questions, please use the raise hand option, which is the third button starting from leave call. If there are no more questions, I would like to request Dr. Deshmukh to say a few words if you are around. Deshmukh? Anyway, it looks like he's not able to hear us. Thank you, Dr. Sanjeeva Nayaka, for the wonderful talk. My last question to you, are you planning to go back to Antarctica, or you would be going to Arctic for more collections in near future?
Yes, certainly I would like to go. The only thing is that the time is really a factor, because Antarctica expedition or the Arctic expedition takes lots of time, because the time you get for real work there in Antarctica is very less because of the harsh climatic condition and other things. The working hours or the working days will be less. The other days in the journey are simply keeping quiet without work is more. So Antarctica would take minimum 3 months, and Arctic again will take 1 month or so. So time is a great challenge for me. I don't have that much of time. So otherwise, I'm very much willing to go.
I'm tempted to ask you, we have studied that lichens can be a good indicator of climate change. So what do you think, what is your hint for the young researchers who might be interested in lichens in extreme environment?
Yes, certainly. Already lichens can be used in different ways for the climate change studies. One thing is that you're just comparing the present data and the previous data. So like if you know that some collections or some species are reported from an area, so you can go to that area again and see whether that lichen is again present in that area. And if you don't see them and that your observation correlates with the other environmental factors, parameters, so you can say that climate change is playing a role there. Or you can set up different kind of experiments there. That is also one possibilities. Second thing is that there are different molecules like these carbon isotopes and other studies you can do, and you see the changes in the lichens, carbon isotopes, and the other isotope studies. You can say that whether lichens are really responding to the climate change or not. If it is responding, how much effect has been done onto the lichens can be studied. Yeah.
Thank you, Dr. Sanjeeva. Dr. Deshmukh, if you are around, I would like to request you to say a few words before I propose the vote of thanks. Dr. Deshmukh.
Yeah, it was very interesting lecture because when I have visited, I could see only crystals lichen over there in both the areas, and they are very colorful. But it was very difficult to pick up even we had tried our level, so we were getting few milligrams. So I think topic is good, and it is for if somebody is doing bioprospecting, that will be useful for pigments as well as for the anti-infectives and anti-cancer compound.
Yeah, certainly, sir. Sure, it can be done. Somebody has to take up such studies and do it.
You can talk to Abhijit Kate and other that group.
Yeah. Already I'm in touch with them.
Yeah, thank you. I would like to take this opportunity to thank Dr. Sanjeeva Nayaka for the wonderful presentation today. And, thank you for the collaboration with the Indian Lichenological Society. Ladies and gentlemen in the audience, you are most welcome to apply for the membership of the Association of Fungal Biologists, and you are also most welcome to apply for the membership of the Indian Lichenological Society. I have shared the links to both the societies, both the organization in the chat box. Thank you, Dr. Sanjeeva Nayaka.
Thank you very much. Again, thanks to you, to the AFB and ILS for the opportunity. Thank you very much.
Yeah, thank you. I would like to thank Dr. Deshmukh for his guidance, his motivation, and for his leadership at the Association of Fungal Biologists. Thank you, Dr. Deshmukh, and I would like to thank the other members of the AFB and ILS for their kind participation today. And ladies and gentlemen in the audience, you have been wonderful. You have been awesome. Thank you for participating in today's talk, especially during the year-end holiday season, actually. And you have a great year ahead, 2025. We would like to see some of you in the next annual conference of Association of Fungal Biologists in December 2025. Thank you very much, ladies and gentlemen. With this, we sign off. Thank you. Bye-bye.
Thank you. Bye.
Thank you.